All right, everybody, we're moving on to chapter 13, where we're going to focus on our divisions of the brain and the cranial nerves as well. So this should be a little bit of a review from the brain dissection and cranial nerve lab. All right, so we know when we did our dissection that if we cut right down the longitudinal fissure to separate the right and left hemisphere, that this is what we're going to look at. So you guys should be able to, as a little bit of a review, take a look at this picture and kind of see how many different structures or divisions can I pick out from the brain. So just like the nervous system, we can divide the brain into some individual divisions or sections and then break it down further from there. So the first and main portion of the brain is going to be the cerebrum. And so the cerebrum is all of the four lobes that make up especially a lot of the gray matter of the brain. So remember in our spinal cord, we saw that butterfly or H shape of gray matter in the internal aspect. On the outside of the spinal cord, we had the white matter. We flip it in the brain. So the cerebrum is a lot of our gray matter outside. And then once we go internal, that's where we're going to find all of our white matter. So the cerebrum is made up of the four lobes. We have the frontal, parietal, temporal, and occipital. And we've said this a couple of times now. If you remember your bones of the skull, our lobes of the cerebrum match up with that. And the cerebrum is just those four lobes and really a fifth if we add in the deep lobe, the insula. So we can't say the cerebrum and the brain are the same thing because when we say the brain, we're really talking about everything on this slide. The cerebrum really refers to those four or five lobes when we add the insula in. The diencephalon is our central portion here that's got our pineal gland or epithalamus, the thalamus, and the hypothalamus. We have the brainstem, and so this is actually organized from inferior to superior. I like thinking of it the other way around just because the midbrain is closest to middle of the brain. The pons is kind of our thickened section here, and then the last portion here is the medulla oblongata. And just another reminder that our brainstem doesn't end and our spinal cord begins. It's a continuous structure. Once our brainstem exits out of the foramen magnum, now we have the spinal cord. And then finally, we have the cerebellum. We call that the little brain. You can see the arbor vitae, the white matter portion there, the tree of life that spreads out there. But remember the cerebellum. And so just to keep you guys oriented, here we have our posterior side and our anterior side. So this would be our frontal lobe, parietal lobe, temporal lobe would be here if we had both halves of the brain, occipital, and so the cerebellum is going to be on the posterior inferior side of our cerebrum. Okay, talking a little bit more about the brainstem itself. And so one thing that you'll see here is the thalamus. Then we'd have the hypothalamus below that, and then we're going to have the midbrain, pons, and medulla. We can see a lot of the cranial nerves and other nerves that are arising from the brainstem. Our brainstem as a whole, we talk about having some of our reflexes and life centers, but we can give some individual jobs to each individual portion of the brainstem. So starting with the medulla, the most inferior part of the brainstem, this has a lot of tracts of the brain. So think to yourself, are tracts white matter or gray matter? And so our tracts are going to be axons and myelinated axons specifically. So they're going to be white matter. Sometimes we call these tracts at this point our pyramids, if you see that word at all. This is where cranial nerve 8 through 12 is going to arise from. You don't necessarily need to memorize those numbers that go with each section. I'm more concerned with you guys paying attention to our functions here. So our medulla oblongata is really where most of our life centers. So our really important reflexes for maintaining homeostasis and keeping us alive, like heart rate, our breathing rate, even stuff like coughing and sneezing that help us get debris out of our respiratory system, swallowing, hiccuping, vomiting, all the different autonomic reflexes that we don't necessarily think about but are super important. Even blood pressure can be controlled here partly, so a lot of important stuff in the medulla, and this is why swelling that's impacting that inferior portion of the brainstem can be really scary. Our pons acts kind of like a middleman or a bridge to the upper brain. We have some more cranial nerves that are now going to branch off of here. It has the pontine nuclei, and so that's what tells us when we're holding our breath, hey man, you got to breathe. And it also is going to initiate rapid eye movement. So rapid eye movement is something that happens especially during sleep and dreams. And so when we dream, this is more of a nice to know than need to know. Most of our body is pretty paralyzed except for our eyes. And so rapid eye movement is associated with our deep sleep. So super important for that. And it's also going to be a bridge from the cerebellum to the frontal lobe. So we'll talk about the lobe's job, but our frontal lobe does a lot of our motor information. And we said the cerebellum helps with balance and coordination. It makes us look like we actually know how to move. So our pons is the middleman relaying that info from the frontal lobe to cerebellum and vice versa. Said the midbrain we remember is the top portion because it's closest to the middle of the brain. This is actually where our cerebral aqueduct travel through. We have a lot more tracks that are ascending and descending traveling through there. And that's also where we get a few more cranial nerves arising. So three, four, and five. Remember, we saw one and two on the underside of the brain itself when we did our dissection. The midbrain is also where the superior and inferior colliculi are found. And so if you remember from our dissection, we kind of pushed the cerebellum inferior and looked through where that space is, and we saw the little baby butt. That was the superior colliculi, and the inferior colliculi is below. So superior goes with our visual reflex, inferior goes with our auditory or hearing reflexes. And so I always use the example when someone calls your name and you turn your head quick without even thinking about it, that's our superior and inferior colliculi or our midbrain running that reaction. The reticular formation is a new structure for us. This is not one that you are responsible for from a lab standpoint, but it's a super important structure. And it's really not just one structure. It's a large group of structures that are located throughout the brainstem. It has both ascending and descending tracks. And so, again, I want you guys to think to yourselves, what would the ascending tracks be related to? What would the descending tracks be related to? Knowing which direction sensory and motor or afferent and efferent information travels. So we know afferent arrives and ascends. So our reticular activating system, our reticular formation, the ascending portion, is going to be related to integrating a lot of our sensory information. So there's a lot of sensory info that travels through different parts of our reticular formation before it goes to the thalamus or the parietal lobe, for example, where a lot of our other sensory centers are. Our descending tracks are going to be motor related, and they go really to the motor neurons of our spinal cord to help us maintain our postural muscles. If you guys remember back from Unit 2 when we talked about the muscles a bit more, or sorry, Unit 3, we said that if we didn't maintain a muscle tone, if our postural muscles or all of our muscles turned all the way off, we'd kind of just slump over. So we need to have that system that's actually talking with our postural muscles and keeping us upright even when we're not thinking about those voluntary movements or standing tall. All right, the cerebellum, sometimes we call the little brain because it's almost like its own little brain attached to the brainstem. It has the transverse fissure because it kind of goes across here. And then this portion, the white, we call the arbor vitae. So the transverse fissure is almost right where we found that fourth ventricle, the little triangle space. That was one of the last spots for our cerebrospinal fluid. And so it helps control gross body movements, meaning larger body movements. It also helps regulate posture along with the reticular activating system, as well as equilibrium, which we'll talk about a little more in our special senses unit next unit. It also helps with fine motor control. And so I always say with the cerebellum helps us make movement look smooth or make our movements look good. So our frontal lobe has a lot of the actual motor information, but our cerebellum helps coordinate all this. So someone with cerebellar damage knows what movement they want to make. It just looks really ugly. So a lot of times they have a hard time with walking. They look kind of like Frankenstein. If you ask them to touch their nose to their finger back and forth, they kind of hit themselves in the face because they have a hard time judging or coordinating that movement. They have a hard time flipping their palms or hands back and forth. So it really coordinates and makes our movements look good. It's kind of putting everything together. It's integrating our movements. So our diencephalon is a deep portion of the brain, so we can't see this when we're looking at the outer portion or the outer part when we can only see the cerebrum. The thalamus makes up a large majority of our diencephalon, a large majority, so 80%. So within the thalamus, we have our sensory relay station. And that's really the big thing to key in on here. So our parietal lobe we'll talk about has a lot of our sensory centers, but all of our sensations go through the thalamus. So our thalamus is also an area where we really can interpret stuff like pain, and we kind of think how what we are sensing, how does that actually make us feel? How does that relate to our emotion? How does that relate to our reactions? So all of our sensory information passes through the thalamus before it gets to its end point. This interthalamic adhesion is just where we actually have the connection. So the third ventricle, another space where we have cerebrospinal fluid, kind of splits the thalamus in half. Just like we have the corpus callosum to connect the right and left sides of the brain, we need a way for that information to still get to all the portions of the thalamus. So the interthalamic adhesion is kind of the bridge or how we keep the thalamus connected, even though that third ventricle passes through that structure. The hypothalamus is our autonomic nervous system, or a big controller for our autonomic nervous system, I should say, influences some of our behavior. And then other stuff like hunger and thirst. This is where temperature information goes. Somewhat influences the sleep-wake cycle, and that goes along with our epithalamus, the pineal gland. So the pineal gland secretes melatonin, which is a hormone that helps govern our sleep-wake cycle. And melatonin uh, is really released in connection to changes in light. So the darker the environment, the more active the pineal gland is, the more melatonin being released. Another real small area of the epithalamus is called the habenula, and this is not something that's super important to us, but can be interesting just because it's very connected to behaviors and addictions. Same thing with the hypothalamus. So that's one that you might want to do your own research on if you're interested in that aspect. Okay, going back to the cerebrum, this is going to be the largest part of our brain. We have a left and a right half of the cerebrum, which is divided by the longitudinal fissure. So some important vocab here listed below for the cerebrum. I said whenever I think of gyri, I say gyridge in my head. So those are the bumps. We have the sulci are the shallower grooves or the depressions that we see along the brain surface. Our cerebral cortex is the gray matter on the outer portion of the brain. The nuclei is going to be gray matter, but deeper inside the brain. And then we have the cerebral medulla, where we get to the white matter. So medulla tends to refer to the middle. So this isn't the brainstem medulla, this is the cerebral medulla. And this is all the white matter that connects our cortex and nuclei. And so this is going to consist of tracts. So remember, axons, myelinated axons are tracts that are going to connect all the different areas of our cortex to each other and to other parts of our central nervous system, like the brainstem and cerebellum and thalamus, and kind of all those pieces we've already discussed. The corpus callosum was one that is on your practical list, and that's our largest bundle of commissural fibers, meaning our connective fibers. So those are white matter, and that's how our left and right hemisphere communicates with each other. A fissure is a really deep groove. So the best example of this was our longitudinal fissure that went down the middle of the brain and separates the right and left half. And our lobes, we technically have five. This is the one that is always forgotten, is the insula or the insular lobe. So we have five lobes that match up with the skull bones if we include the insula. So frontal, parietal, occipital, temporal, those are our skull bones and our lobes. And then we add the insular lobe as our fifth lobe, which is a very deep structure. Here's another chance for you guys just to practice a little bit with your labeling, so keep that in mind. Okay, so we mentioned the meninges in our last chapter, so we'll go over those briefly again. But the cranium itself is our skull. So when you say the cranium, you're referring specifically to our skull bones. We had the cranial bones that make up the skull, and we had the facial bones that kind of make up the rest of the head and face. The meninges are our protective covering here, and these are going from superficial to deep. And then cerebrospinal fluid, remember, we only find in the central nervous system, and it's named for where we find it, the cerebrum and our spinal cord. So remember, the word ventricle just tells us it's a space in the brain, and our ventricles are all connected to one another. So we looked at a few different spots in our dissection, but keep in mind, those are all one large connected series that are going to allow cerebrospinal fluid, or CSF, to travel throughout the brain, through the spinal cord, and then cycle back up. And so remember, we don't just get CSF out of thin air. It doesn't just poof into our ventricles. We have blood plasma that is filtered by our ependymal cells. So that was one of our glial cells. It has two jobs here. So that CSF provides protective cushioning around the brain and spinal cord. In football players, obviously concussions has become a really big topic, and they've definitely shown pretty clearly in research that being dehydrated is a big risk factor for having a more severe concussion, because that typically means we're going to have a lower volume of CSF as well. And obviously, it's a really important for nutrient exchange. So there's glucose and other nutrients within that cerebrospinal fluid that are going to help keep the brain healthy, keep our glial cells healthy as well. So we said this earlier, it's filtered blood plasma. And so our choroid plexuses are the combo of our ependymal cells and our capillaries, the blood vessels that go with them. So our capillaries filter out the plasma portion of the blood, which is the liquid portion. It's mostly water, and that becomes our CSF. This is one you definitely want to star on your slide. You'll see that again for sure. Make sure you know that order. So we start in the larger lateral ventricle. We then go to the third ventricle. The cerebral aqueduct, I would say, is kind of our water slide from the third to the fourth. Then we go to our subarachnoid space. So that's between our arachnoid mater and our pia mater. And then it travels into the central canal of the spinal cord. That was the kind of the small pinhole that we saw in the very center of the spinal cord. We can see that on our spinal cord models in the lab as well. So here's an illustration of that. This is another picture that you should kind of star. So you should know blue here is our lateral ventricles. So this is our largest starting point. We have our third ventricle here in green. The cerebral aqueduct is our water slide down to the fourth ventricle. And then we're going to go through the subarachnoid space to get through the cerebrum. And it's our central canal that is going to bring that CSF into the spinal cord. All right, the blood-brain barrier we talked about a little bit in chapter 11 when we talked about our glial cells. And remember, this is our astrocytes that are responsible for forming this. So capillaries are the smallest vessels. This is where we actually exchange material. So it's actually the capillary network plus our astrocytes on top of these capillaries that form really tight junctions and allow us to make this brain barrier or security system for the brain. The blood-brain barrier encloses all parts of the brain except for the hypothalamus. That's the only spot that is exposed. The blood-brain barrier, besides being very protective, also poses a challenge for us from a medical standpoint because we have to be able to get a medication into the brain if we're going to treat it there. So the blood-brain barrier only lets certain substances in. So oftentimes we need to package medication with another medication to help us get through this barrier. So this is part of the reason why something like Alzheimer's, Parkinson's, epilepsy, cancers of the brain are difficult to treat because we're really limited in what materials we can actually get into the blood of the brain itself. So this is a nice table to look at in terms of what do we let in through the blood-brain barrier? What are we always keeping out? So of course, water, nutrients, oxygen, other gases. For example, carbon dioxide has to be able to get in and out. Glucose because we need a whole lot of energy to keep our brain going. And then the last two are the ones that seem a little counterintuitive. So nicotine, I think most of us know, is contained in substances like tobacco, cigarettes, vaping, chewing tobacco, pouches, Zins, whatever it may be. So nicotine itself can be addictive, but it's not bad for our brain. It's actually neuroprotective, and there's some research showing that some nicotine actually influences or improves brain function. That's not to say go out and start using nicotine, but it's not a bad thing for our brain. It's all the other stuff that nicotine is packaged with that causes the issues. This word ethanol is really the way to say alcohol. So once alcohol is processed in the body, it becomes ethanol, and that can also get into the brain. All right, you guys do not need to memorize this picture, but the table that we look at next that shows all of our different functions you should know. So we looked at the olfactory bulbs as well as our optic nerve and optic chiasm. So you only need to identify those ones that we find right on your list. This is really where you should focus your studying when it comes to the cranial nerves. Remember, there's no other cranial nerves on your lab practical itself except for those couple that we just talked about in the brain dissection. One thing I want to point out here is you'll see some of our cranial nerves do sensory only, like our olfactory and optic nerve. Some of our cranial nerves are motor only, like our oculomotor nerve. And then some of our cranial nerves do some of both. For example, our trigeminal and facial nerve have some muscles that they influence of the muscles of mastication here, that are chewing muscles, but it also does sensation for our face, mouth, and nose. Same thing with our facial nerve. The sensory half does taste for the anterior two-thirds of the tongue, and the motor half does all of our facial expressions and helps release and produce saliva and tears. So we call that a mixed cranial nerve. So you can have sensory, motor, or mixed. You'll notice we always label our cranial nerves or number our cranial nerves with a Roman numeral. You should never see those written with Arabic numbers. A lot of the names of the cranial nerves also help you with what their function is. So use the names to help you remember these functions. Oh, sorry. And in terms of memorizing your order from 1 to 12, there is a ton of different sayings out there. You also can find sayings that help you remember which ones are sensory, motor, or mixed. We're not going to necessarily test you directly on that aspect, but you should be able to match up your functions. One great way to do this is kind of mix these up and be able to put your names back in and put them back in order, and you're in a really good spot. This is one of the first kind of checkouts or practical skills that you'll be tested on when you're in the nursing program. So it's great to get a head start on these. The way that we used to remember them in PT school in terms of the names themselves were O, to touch and feel, very good velvet, ah, happy. So that's the first letter of all the different nerves. And I know it seems silly, but I still remember it now. And you can find a ton of different variations on that. So pick whatever one works best for you. And I will see you in chapter 14 where we kind of put everything together.